So next type of method of measuring the cardiac output is dye dilution method. So as I already said, you'll be having five types of method for measuring the cardiac output. So fixed method, dye dilution method, indicator dilution method, thermal dilution, and impedance change. So from these five type, already two we had seen, that is fixed method and indicator dilution method. So next we are going to see the dye dilution method. Okay. So a common method of clinically measuring cardiac output is to use a colored dye, indocyanamine green. Okay, here we are going to use the type of dye that is cardio green. Okay, to measure the cardiac output. So here we are going to meet the necessary requirements for an indicator, that is the dye is available as a liquid, that is diluted in isotonic saline and injected directly through a catheter, usually into an pulmonary artery. That means the dye indocyanine green dye will be in the appearance of liquid, which will be mixed or diluted with isotonic saline water. Okay, so after that only are going to inject into the pulmonary arteries of the heart. Okay, through the catheter. Okay, using catheter you are going to apply a dye to the heart, pulmonary artery of the heart. So about 50 percentage of the dye is excreted by the kidneys in the first 10 minutes. Okay, so repeat determinations are possible. That means the 50 percentage of the dye will be excreted by the kidney for the first 5 minutes. After that it may repeat or may not. Okay, so the plot of the curve for concentration versus time is obtained from a constant flow pump, which draws blood from a catheter placed in the femoral or brachial artery. So the concentration of the um, dye can be plotted, okay, using the curve with respect to the time period. Okay, it will be that is nothing but the blood which we are going to draw from the catheter which is placed in the arteries. Okay, so the constant flow in the arteries can be measured by um, using this concentration of dye. Okay, next, blood is drawn through a calorimeter cuvette which continuously measures the concentration of dye using the principle of absorption photometry. So calorimeter cuvette, okay, is an instrument which going to measure the concentration of dye. Okay, how it going to measure means by using the principle of absorption. So how much it is absorbing? Okay, absorbing the dye which is received from the blood, heart of the blood, sorry, blood of the heart. Okay, so nowadays, next method, thermodilution method. This is a dye method. So we are going to inject dye in the blood of heart. Okay. So after injecting, you are going to take the blood for the measurement of cardiac output. So from the blood, you will be finding the concentration of dye. Okay. So on finding the concentration of dye, you may able to know the cardiac output. Okay, which will be respective to each other. That means it will be proportional to the concentration of dye. Okay, cardiac output will be yeah, yeah, proportional, directly proportional to the concentration of dye. So using the dye, you can measure the cardiac output value. So next method will be thermodilution. So thermodilution method is recently adapted for measuring the cardiac output. So maximum we are using this thermodilution method. So a bolus of about 10 ml of 5 percentage dextrose in water at room temperature is injected as a thermal indicator into right atrium. So bolus is nothing but we are going to mix the bolus with water, okay, at room temperature, and then we are going to inject it to the blood of heart in the right atrium. Okay, I think you all know atrium, ventriculus are the parts of heart. Okay, right atrium, we are going to apply this thermal indicator, that is bolus. So after mixing, it detected in the pulmonary artery. We are going to enter the indicator, thermal indicator in the right atrium, but we are going to take out, okay, or detect from the pulmonary artery of the heart. Okay, so by keeping thermistor at the tip of a miniature catheter probe, so using catheter only you can able to measure the blood flow in the heart. So in the catheter probe, you are going to mount the one more thing, that is thermistor. Okay, so thermistor is here used for detecting the mixed thermal indicator in the heart blood. Okay, next, the temperature difference between the injector temperature and the circulating blood temperature in the pulmonary artery is measured. So before injecting, okay, you'll be measured one temperature. So after injecting that indicator, the blood will have some other different temperature. Okay, so these two temperature difference will be measured after detecting the blood at the pulmonary artery. Okay, the reduction in temperature is integrated with respect to time. So the change in temperature will be going to measured with respect to the time period, and the meter reads the cardiac output. So this will be giving the cardiac output reading. Okay, so change in temperature of the blood while entering the temperature indicator, after leaving, after mixing that dye, sorry, indicator with the blood, you will be getting some temperature in the blood. Okay, these two temperature difference value will be measured, that will be proportional to the cardiac output. Okay, so cardiac output is nothing but the blood flow for a minute. Okay, the measurement of blood flow in the heart for a minute is said to be cardiac output. So next we are going to see the impedance change method. So by the name itself, by changing the impedance value, change of impedance value will give cardiac output. So by the impedance method, the cardiac output can be determined electronically. Here we are going to use L probe. L probe method going to adapt here. The electrode pair 1 and 4 will be used as a current electrode. 2 and 3 electrode pair will be used as a voltage electrode, that is used to pick the voltage across your thorax. Okay, what is thorax? So when a consistent current at 100 hertz, 100 kilohertz is applied between 1 and 4, that is current electrode, you are going to apply the current value 100 kilohertz. Okay, the impedance value 25, and this will be diminished to with each systole. Each systole means loved up. So for contract, expand, contract, expand. So systole, diastole. Okay. So while doing systole, you are going to apply the 100 kilohertz current to the 1 and 4 electrode. That is current electrode with impedance value 25 ohm. Okay. So the voltage signal due to changes in impedance value will be amplified and demodulated to obtain Z. So on looking the diagram, you will be known. See here you are going to use four electrode, one, two, see one, two, three, four electrodes. Okay, four electrodes are used to find out the impedance change. So here you are going to use the four electrodes in the human body to place on the surface of the human body. But these electrodes will be further connected to the electronic components. Okay, you will be using a differentiator, amplifier, and then few resistor value. Okay, see this is nothing but oscillator, two resistor, amplifier, This the entire circuit with omp amp will be called as differentiator. Okay, so this A1 is nothing but amplifier as well as demodulator. Okay, so the current value we are going to apply to the 1 and 4 electrodes. Okay, so after applying the current value to the 1 and 4 electrodes, you will be collecting some potential value in the 2 and 3 electrode. Okay, this will be given to the amplifier and demodulator where it is amplified. The voltage which is drawn from the 2 and 3 electrode will be amplified and demodulated in the A1. Okay, A1 device, and then it will be given to the differentiator. Okay, so differentiator will do the some differentiation value. Okay, of that biosignal which is taken from the heart, and then it will be provided as a D signal. Okay, that is the point they given here. So when heart muscle contracts, so contracts means when it is unfilling the blood. Okay, so removing all the blood, that is said to be contracts. Blood is ejected from the ventricles, and a pulse of pressure is transmitted through the circulatory system. Okay, so when your heart muscle gets unfilled, that is contracts, blood will be ejected from the ventricle. Okay, leave the ventricle, and then a pulse will be created. Okay, that is due to pressure there will be a pulse. Okay, pulse will be created due to pressure that is transmitted through all the parts of the body. Okay, or circulatory system is nothing but your heart going to circulate all the blood. Okay, to the all the parts of the human body. So this pulse can be measured at various point. You can measure this pulse at various point. We can sense the pulse by 1 minute. Hello, ma'am. Ma'am. ஆமா மேம். நான் சர்ச் பண்ணி பார்க்கலாம் மேம். மெயில்ல இருக்கும் மேக்ஸிமம். சரிங்க மேம். இந்த கிளாஸ் இந்த ஹவர் போயிட்டு இருக்கு மேம். 2 மணிக்கு மேல அனுப்புறேன் மேம். சரிங்க மேம். Okay. So this pulse can be measured at various points. We can sense the pulse by placing our fingertip over the radial artery in the wrist. So you can sense this type of pulse by your finger itself in the by keeping the radial artery in the wrist. டாக்டர்லாம் பல்ஸ் பிடிச்சி பார்ப்பாங்க இல்லையா in the hand. Okay, wrist, wrist of the hand. So this is nothing but they are checking the heartbeat level only by placing the their finger in the wrist of the hand. Okay, so by this means also you can check the heartbeat of the human body. Okay, so this pulse travels at the speed of 5 to 15 meter per second. So the photoelectric method is commonly employed to measure the pulse. So for measuring the pulse, you are going to apply the photoelectric method. That is nothing but which will be converting the electrical energy into light energy. Okay. So pulse measurement types are transmitted method and reflectance method. Two methods for measuring the pulse. So pulse measurement நாளைக்கு பார்க்கலாம். It's enough. Impedance change method ஓட போடும். Pulse measurement is next topic. Okay. It's enough. So simply we can say by using the change of impedance value between the 1 and 4 electrodes, which is the current electrode. So due to that you will be getting a voltage at the 2 and 3 electrode. This electrode will measure the biosignal from the heart. This will be given to the amplifier and demodulator, and then further to differentiator to get the D signal. So D signal is nothing but your cardiac output. Okay, that's all.